All right, today we're going to discuss staining procedures in the lab to visually distinguish between different types of bacteria. All right, so a general overview of staining. Stains. And when we stain, we use certain specific dyes that bind to specific molecules or structures of interest within a cell. So we'll apply those dyes, and then we'll look at the cells under a microscope so that we can visualize those specific structures. So you can see on the right side, we stained green. We used a green dye that specifically binds to like the cytoskeleton, so you can see all those filaments in the cell. And then we have for example, we have a blue dye that binds to the DNA, so you can see those nuclei within each cell there. So you can just visually look at all these structures that we want to see using staining. So there's a lot of different complicated stains, but we're going to look at the two most essential ones that are used in microbiology, and they're going to be used to categorize bacteria into two groups. So let's go over some terminology before we get into this. So we have two general broad categories of stain. One is a simple stain and one is a differential stain. So a simple stain, you're only going to use one dye. So for example, we have a dye called methylene blue, and then these stains, they're not going to be super detailed. You're not going to get all that contrast between different things. You're only just going to get a general more, I guess the intensity of things is going to be more apparent. So if the cell's all clear, you can't really see things clearly. If you make it blue, then you can see different structures more clearly, but they aren't differentiated by color here. And then within that, you have a direct stain where you actually stain the cells themselves. Then the negative stain where you stain the surroundings. And the direct stain is going to be the one that we'll always look at. We're not going to look at any negative stains for our purposes. Okay, so then the second type is what's called a differential stain. These are going to be the ones we focus on in the next few slides. So here you're going to use what's called a primary stain. And after you apply that, you're going to wash it away with what's called a decolorizer, and then you're going to stain again with a second stain that's called a counter stain. So let's get into how all of this works. So we'll start with the Gram stain, and this is you'll see this everywhere if you ever do microbiology in the future. This is kind of the most basic stain that is a staple of the field. So Gram staining, it's a procedure that we can use to visually distinguish between gram-positive and gram-negative bacteria. So if you remember, we went over gram-positive, gram-negative when we talked about our bacterial structures. So reminder, gram-positive, you have a thick cell wall. Let me get my pen here. Yeah, so you have this thick cell wall made out of peptidoglycan, which is that long sugar polymer that's connected in between using those amino acid / protein units, right? So we have big cell wall, we have one membrane here, whereas in gram-negative bacteria, we have two membranes, the inner and outer membrane. Then we have a very small cell wall in between that has low peptidoglycan content here. So let's see how, okay, so the Gram stain is going to allow us to visually distinguish between these two types just looking under the microscope. So let's see how this works. All right, so to start, you're always going to start your stain by doing heat fixation. So what this means is that once you have your culture of cells, so let's say you have a petri dish, or you have some tube, right, with all your cells, all your bacterial cells, you're going to take a little bit out. You're going to scoop them out. You're going to place them onto a microscope slide, and then you're going to place the slide over a heat source like a Bunsen burner, as you can see here, Bunsen burner. What that's going to do is that it'll adhere all the cells you collected onto the slide so that they don't get washed off when we're applying the dye or when we're rinsing off the dyes later on. All right, so next step, you're going to flood the region that has the cells, this region, with our primary stain, which is called crystal violet. And as you can expect, it's a purple dye. And we'll let that sit for a minute so that the cells absorb all the crystal violet, and then we'll rinse it off with water. Yeah, so crystal violet, and it's a purple dye that specifically binds to peptidoglycan. So it's going to be present in all the cell walls of the bacteria in the sample initially. And we'll see how that'll change in a second. Yeah, so next, after we do that, we're going to flood the slide again. We're going to cover the slide. You can see on this first step here. We're going to cover with iodine. And this is what we call a mordant. So a mordant is something that really allows the initial dye to bind permanently. So basically, after we add the iodine, it'll help the peptidoglycan really stay embedded into those cell walls. So we're going to flood this slide for 1 minute, and then we'll rinse off with water again. So like I said, iodine will bind the crystal violet and affix the dye into the cell walls. Yes. All right, so after we do that, now we're going to do the decolorization steps. So we're going to add either acetone, ethanol, or acid alcohol. Ethanol is just like pretty much normal alcohol in the lab. Acetone is like, you guys know like nail polish remover, right? It's that chemical. Acid alcohol is just a mix of like ethanol and some like hydrochloric acid. So what this is going to do is we'll put it on the slide briefly for 3 seconds and then immediately rinse it off with water. This decolorizer will wash away the crystal violet, but not in all the cells. So, and why is that the case? So remember that the gram-positive cells, right, G positive, have the membrane, and then they have the thick cell wall here, right? So when we put the crystal violet in, it's all stuck in this like thick membrane, thick dense cell wall. So even when we add this like ethanol, acetone, acid alcohol to wash it away, it's not going to get washed away because it's so tightly confined within that big cell wall, okay? Now the difference is that for gram-negative cells, right, we have membrane, then we have a thin cell wall, and then we have another membrane. So since this membrane's pretty thin, right, the crystal violet, it's going to bind here, it's going to bind here, it's going to here. But when we add the decolorizer, right, there's not that much peptidoglycan to bind to, so these guys, they can just get washed away pretty easily, right? So essentially what that's going to do is that after we add the decolorizer, we're going to have all the gram-positive bacteria be purple because the dye is still stuck. Then we're going to have all the gram-negative bacteria become clear or colorless because all the dye has been washed out since they don't have that much peptidoglycan for the dye to bind to. You can see that on the right here. These guys are gram-negative, and then these guys are gram-positive here. So finally what we're going to do is we're going to flood the slide with safranin, and this is our counter stain, our second stain. So we're going to do that for 1 minute and then rinse away again. So what's going to happen is that safranin, it wants to get into all the cells, but the gram-positive cells are already purple and like filled with dye. So this red dye, the safranin red dye, won't get into those. So the gram-positive will stay purple. And then the ones that just turn clear because of the decolorizer, the gram-negative, right? The gram-negative, they go from clear to red because the safranin will selectively go into those cells since they don't have any dye. Yeah, so once this step is done, as I said, all the G plus gram-positive cells will be purple, all G minus will be red/pink. And this is very important because you'll probably be given a microscope image, and then it'll give you different colors of cells and tell you and ask you to tell which one's gram-positive, gram-negative, etc. So and then the final step would just be to dry a slide on what's called babulous paper. It's just paper that absorbs water very well since we rinsed here, right? So we're just going to absorb all the water away, and then we'll stick that under 1000x oil immersion, right, with the bright field microscope like we talked about in the microscope lecture. Yes. So here's an example image of what you might see under the microscope. So you see all these. If you remember, these are Staphylococcus, right? These clusters of spherical bacteria, and then these are bacillus bacteria that are elongated shapes, right? So the bacillus, these guys are gram-negative here, whereas the cocci are cocci. I don't know how you say it. Yeah, but those guys are gram-positive here. And you can see like the scale bar. We did all the size estimation stuff last time. So yeah. Yeah, so recap, these are like the key pieces of information here. Remember, primary stain crystal violet, the mordant iodine helps the crystal violet to bind. The decolorizer removes the dye only from the gram-negative cells, and then the counter stain safranin stains only the gram-negative cells, so you're left with this final result here, and this is a pretty good diagram for you guys to review. Yeah, so I linked a video here that, and this procedure is slightly different, but has the same overall steps. So I highly encourage you to go watch this because I don't know about at like Jinx or like local tournaments, but at the state tournament last year, they made us perform a gram stain like live. So I would highly recommend that you look through this so you get a sense of what's going on. Yeah, so there are some exceptions with gram stains because the gram stain depends on the fact that the crystal violet primary stain is binding to the cell wall, the peptidic glycan. So some bacteria, like for example Mycoplasma, these guys don't have a cell wall at all, so they're not going to get stained properly with the crystal violet. Then chlamydia and then rickettsia. These are very extremely small bacteria that have trouble taking up this dye as well. And these guys are actually both, well, I'll write parasite. You can think of, I guess all of bacteria as a parasite, but you can think of them as parasites specifically because they can only, these bacteria can only survive within the host cells. So they don't like float around on their own. They're always within a host, which is why they're very small. And then Mycobacterium, this is another one that it can resist decolorization. So after you get crystal violet in, it won't get washed out, even though it's gram-negative, because it has a very lipid-rich cell membrane that the dye has a hard time getting removed from. So it'll stay purple, even though it's a gram-negative here. All right, so let's go into our next procedure here. This is the acid-fast stain. So the acid-fast stain, it's a procedure that's used to specifically identify bacteria that have what's called mycolic acid in their cell walls. And this includes Mycobacterium and Nocardia. These are just names of the genuses of the categorizations of bacteria that fall under this classification. And if you remember Mycobacterium, it's the one here. So it has that waxy lipid-rich cell membrane. And so mycolic acid is what makes it that way. And it also has, you may not need to know this, but I would including it just in case, arabinogalactin. This is another sugar polymer that's also found in the cell walls here. So you can see this diagram of the acid-fast bacteria cell structures on the wall on the right. So again, this is a differential stain. So we'll have the primary stain, mordant, counter stain, primary stain, mordant, decolorizer, then the counter stain. So the reason this stain is useful is because these, specifically Mycobacterium, this group of bacteria, they are very good disease causers. So for example, tuberculosis and leprosy are both caused by these bacteria. So if you have like a sample of human tissue from like a patient, then you can do this acid-fast stain on that sample. And then if you find acid-fast like positive bacteria, it probably means that patient might have like tuberculosis or leprosy, depending on their symptoms as well. And then the word acid fastness, it refers to the bacterium's ability to resist decolorization by acid alcohol. So like what we talked about here, right? Because that mycolic acid prevents the purple dye from coming out of the cell, so it stays in there. Right, so let's go through the procedure. Pretty similar steps, just different components within each one. So yeah, first we'll heat fix like before. So we'll use Bunsen burner to adhere those cells to the slide so that they don't fall off later. Next, we're gonna adhere. I'm not exactly sure why they do this. I would suspect maybe it's because the dye is very strong, so that they don't want it. They don't want it to directly spill onto the cells, but I'm not sure. But apparently you have to put a paper towel or filter paper on top of the specimen and then pour the primary stain on top of that. So the primary stain is what we call carbofuscin here. And this is a red primary stain, as you can see on the right here. So again, this guy will get into all the cells, just like the crystal violet. So all the cells will be stained red. And then what we'll do is that instead of using iodine mordant like last time, we're just going to use heat as our mordant here. So we place the slide over what's called a steam bath. So basically we have water and then boiling water, steam coming up under the slide. We'll use that heat to allow the dye to be permanently binded pretty much to the sample here. So yes, the key is that heat acts as the mordant, not a chemical. Right. Okay, so after we do that, we'll let that sit, and then we'll do the decolorization step, right? We're going to use acid alcohol, and then we're going to decolorize for 10 to 15 seconds, then rinse with water. And what's going to happen is that the acid-fast bacteria, right, they have, remember, the mycolic acid in their cell walls, and carbofuscin will actually bind specifically to mycolic acid. So once you do the mordant and the fixation and everything, the carbofuscin will stay in the acid-fast bacteria, but it won't be permanently bound to the non-acid-fast bacteria because they don't have mycolic acid. So in this step, the non-acid-fast guys are just going to get their primary stain just washed out of them. So these guys are clear here. Yeah, so as you can see, we have the red that are acid-fast, and then we have these clear cells that are not acid-fast. Yep, and then we'll do the same final step. We'll flood the slide again with a counter stain. In this case, we'll use methylene blue, and this is just a blue dye. So all the cells that are clear, all the non-acid-fast bacteria will now turn blue. Then the acid-fast bacteria retain their red color, like similar mechanism to how we talked about in the Gram stain. Excuse me. So as you can see here, now it looks similar to Gram stain, right? But they'll tell you which procedure they used. So here, the red cells, right? They would be acid-fast positive. They have the carbofuscin still in their cells. Then these blue guys, they're going to be the stain with methylene blue. So they are acid-fast, non-acid-fast, or acid-fast negative. So recap, primary stain, carbofuscin, red. The mordant, we use heat instead of iodine. Decolorizer, acid alcohol. And here we specifically have to use acid alcohol because this is kind of the strongest decolorizer, and this carbofuscin dye is very strong. So we need a strong decolorizer to take it out of the cells. Then finally our counterstain, methylene blue, which is blue. And again, I have a video demonstration of this. This one, I think you can watch if you want, but I highly doubt that they would, maybe not highly doubt, I doubt that they'd make you do this specific one. If they make you do a stain, it's probably going to be the Gram stain. So feel free to watch this if you want to see a real demo of what's going on here.